Podcast is intended to cover the lecture that we missed right before spring break. This was Module 9 that covered experimental design and SULAB research. So just as we normally do, I'll go ahead and cover very briefly the assignments and schedule, what's been happening in the lab. I'll also cover some material that people seem to struggle with a little bit on the first midterm, and then we'll talk about experimental design and the SULAB research. The grades have continued to be very high. The average on Model 8, which covered Drosophila genetics 3, was 91%. Looks like we're doing really fantastic. The two modules that were due on April 3rd were modules 9 and 10. Module 11 will be due on Wednesday, April 10th at 12 o'clock. And as a reminder, we're going to have midterm number 2 on Wednesday, April 17th, also at 12 o'clock. The rooms for the midterm number 2 will be posted on Canvas in the announcements, so please keep an eye open for that as the date approaches. In the lab, what we're planning to do is to set up population cages with very young adult flies with the intention of then collecting the embryos over break for you so that when you come back, you'll have third instar larvae ready for you. We're going to do the best we can to collect as many third instar as larvae and hope to get all larvae into your vials the week after break. Your independent compounds are going to go into food, just as they have with the library. And remember that we're going to be treating the larvae with 2,500 rad rather than 4,000. A lot of the publications that Tintin Sue publishes uses 4,000 rad, but we're going to use 2,500 because in our hands, when we did the radiation titration earlier this semester, we found that 2,500 rad will be sufficient to kill about 50% of the larvae, leaving us some room to be able to see death caused by the compounds that you're testing. On Wednesday, April 24th, this is also a reminder that the Cure Symposium will be held at 5:30. You'll need to be over at the University Memorial Center at 5 p.m. We'll have food and they'll start serving it at 5 o'clock. But you want to get there early to sign in with your TAs, to get situated at your poster because the poster session will begin at 5:30. We've been developing the poster for some time now, and this will continue through the end of the semester. You had your proposal due on March 13th, which will be graded as soon as possible. March 20th would have been the peer review, and you'll get those back as well so that you can see how other people propose the validity of your experiment. You'll be meeting with me or Katie Franks the week of March 20th, and then we'll have you submit drafts of both your methods section and the introduction to get some edits and feedback on what's going on the poster. The deadline for submitting your poster file will be April 18th. This is a Thursday, and it should be uploaded to Canvas by the end of the day. The posters are due to the printer at noon on Friday, and I want to be able to look at all of the posters before we submit them for printing. Now, this deadline is only for printing. It's not for a grade. So if you choose to print it yourself, this is completely fine. It's just the last day for submitting so that we print it for you is April 18th. And again, the symposium is on the 24th. Now, we've covered a lot of material so far, so we're not going to go all the way back to the beginning of the semester, but let's just highlight some things that people struggled with a bit on the first midterm. We are treating our larvae with ionizing radiation, and by definition, ionizing radiation causes DNA damage. Now, the manner in which it does this is both direct and indirect. So the radiation itself can hit DNA and cause double-strand breaks. In most cases, in human cells anyway, when you hit DNA within a mammalian cell and you cause a sufficient amount of double-stranded or single-stranded breaks, it leads to cell death. And this is ideal when you're looking at treating for cancer because you want the cells to die. Another way that the cells can die is through indirect damage caused by the radiation, which hits water. And the water gets split and creates reactive oxygen species. These are very reactive species that affect the stability of the DNA and cause, again, double strand breaks or single strand breaks, leading to cell death. But you should understand that the ionizing radiation that we use, or gamma radiation, causes cell death in two different ways. Another thing that seemed to confuse people was the number of chromosomes that the flies have. So every cell in a fly has two copies of the four chromosomes that contain the genome of the fly. In this image, what you're looking at are the four chromosomes, the sex chromosomes, which are chromosome number 1, chromosomes number 2 and 3, and chromosome number 4. The sex chromosomes are telocentric. In other words, when they're paired, they are connected at the ends. And chromosomes 2 and 3 are metacentric. The exam was asking you to visually identify the chromosomes, and you could have done this through knowing where the chromosomes are connected to each other. So make sure you know this for the second midterm that's coming up. Another thing that seemed to be a little bit confusing was embryo development and maternal effects. So the thing that seems to be a bit confusing is if you have a fertilized egg, you have a nucleus. And it has chromosomes that are both from the mother and the father fly. The nucleus divides, so you end up with many, many nuclei. But it isn't until much later in development that these nuclei are able to support the embryo. In other words, this embryo is really focusing a lot of energy on maintaining or creating polarity, what's top, what's bottom, what's front, what's back, really basic things. And in addition to that, focusing a lot of energy on cell division. Many of the genes that are required to support this kind of development are not yet expressed within these cells, but they are expressed in the mother. So in order for this embryo to make it from here to here, when it's really self-sufficient, it needs the mother to supply either proteins or RNA into this embryo, into the egg, to support what's happening with the cells as they're beginning to develop. And these are maternal effects genes, the ones that are supporting the embryo before it can support itself. At this point, zygotic genes begin to take over. In other words, the mother's RNA and proteins are no longer needed because this embryo can make them itself. Just another review on balancer chromosomes. Everyone seemed to understand this pretty well, but these are the three qualities that you should really keep in mind when thinking about balancer chromosomes. That they are genetically engineered. They're not naturally occurring chromosomes. The genes are there. In other words, there is genetic material on the balancer chromosome, but many of them are inverted. And because they're inverted, crossing over doesn't occur. And what this does is it limits genetic diversity in our population. This is exactly what we want because we don't want to increase genetic diversity by allowing crossing over, because that might disturb our our transgenic or our transgenics that we're trying to maintain in our population. The purpose of using the balancer chromosomes is to eliminate the possibility of wild types. If you're not clear on why this is, let us know and we can give you some Punnett squares to do. So hopefully you can see that over time, if we do nothing, then the wild types will take over because they're more fertile. This balancer chromosome also allows us to distinguish between the homozygous and heterozygous mutants. We're interested in the ones that do not glow because the ones that do not glow contain two copies of mutated grapes. And this is the fly that's going to be most susceptible to radiation treatment. So what I want to talk about next is experimental design. When viewing this, you will have already submitted your proposal, but hopefully this will give you some ideas of the things that you should be thinking about as you're refining your experiments. So just a little bit of history to begin. You might not recognize what this is, this disease, but this is a depiction of scurvy. Scurvy occurs as the result of vitamin C deficiency. It was observed in the 1700s when there were very long boat trips, and people were coming off the boat looking like this. If you can imagine family members wondering what the heck is going on with these people. Sir James Lind decided, well, let me see what might be causing this, and he suspected it was a lack of fruit and vegetables. So what he did is he did a controlled study, the very first documented control study, where he put people on a ship and he gave some of them fruits and vegetables and some people no fruits and vegetables. And what he observed on the other end after the trip was people who got fruits and vegetables did not look like this, did not develop scurvy. And that led later on to the idea that citrus fruits and even later than that, vitamin C will prevent this disease from occurring. What's interesting, though, is despite the power of identifying a treatment for scurvy, the idea of experimental design, the way that we think about it today, didn't actually catch on until much later. Experiments performed by Fisher in the 1920s were really the first ones to be published in a way that we recognize. They included all of the basic principles of experimental design, including controls, randomization, and analysis of variance. And this is a statistical method that he developed to determine whether there are meaningful differences among many different groups. He did this in crops, and what he did is he used different crops that were exposed to different chemicals that he felt were good fertilizers. And he had ones that didn't receive fertilizer, which he called basal conditions, and ones that had known fertilizer. And the idea was that this would hopefully improve agricultural practices, and indeed they did. We still use many of these methods today. The basic principles of experimental design involve exactly what you're doing now when you're designing your experiments. The first thing we're doing is we're formulating a question. You don't want to go into a lab and perform science as a fishing expedition. This is a term that's used when you just go in and perform experiments to see what happens. Really, to have something, to get an experiment to give you an answer that's meaningful, you really have to have a very clear idea of what it is that you're interested in understanding. So in our experiments, the question is, which compounds could represent novel chemotherapies? Put a different way, you would then extend this initial question into forming a hypothesis. Now, this is really important. A hypothesis is a single statement usually, and it's a testable statement. So if you had, say, orange juice and you were interested in understanding whether it could be a potential chemotherapy, what you do is form a hypothesis about what you think it might do. Now, in all of your cases, the things that you're considering in your independent projects are based on other people's experimentations or anecdotal evidence suggesting that your compounds may be useful as a chemotherapy. So your hypothesis in that case would be orange juice and you would probably specify a dose, will kill Drosophila melanogaster larvae at a rate higher than the negative control. That's something that you can directly test. A hypothesis that isn't very good is one that includes things that you cannot test. So you may very well think that orange juice is a cure for cancer, but a hypothesis that's testable in our lab could not be orange juice is a chemotherapy that will treat head and neck cancers in people and improve lifespan. That is not a hypothesis that's appropriate for our lab. The thing that's appropriate for our lab is what you're actually testing. And what we're doing is we're testing to see if your compounds will kill larvae in a model of head and neck cancers. You always want to include a comparison or a control, and we've been doing that all along. The positive control in our case has always been colchicine. Colchicine is a chemotherapy that we know will kill larvae. We know that it's worked in head and neck cancers in the past, although it's very toxic. The positive control allows you to know when you view your data that you're capable of identifying a potential chemotherapy. If your positive control fails, if colchicine does not kill your larvae, you might question whether the experiment was done correctly. The negative control is just to make sure that anything else in the background that you're not interested in is not the thing producing the effect. So in our case, we've always used the solvent. Our compounds usually come as powders with a given molecular weight. You then suspend those compounds into some molarity in a solution. And the solution itself, whether it's water or chloroform or DMSO, is probably going to have an effect itself on the larvae. So if we think about chloroform or DMSO,
these things might be toxic to the flies. So, what if you tested a number of different compounds that are suspended in DMSO and found them all to be hits? Wouldn't that be cool? Except that when you go to the next step and somebody does the right controls, what they'll find is that maybe it was just the DMSO and not the compound at all. You want to separate out that background. Some people have been asking about water, why water would be a negative control and why you need it. Water, it doesn't do anything, right? But in our case, our compounds are suspended in food. And if you make water represent some proportion of the food, if you think about that, you're decreasing the concentration of nutrients. So what if you added 300 microliters into 2.7 milliliters of food? You're decreasing the amount of nutrients available to the flies by 10%. And that itself can be toxic to the larvae. Replication is extremely important because this is an ongoing problem in science where there may have just been some silly mistake that you made that made the experiment work. You have to repeat it or have someone else repeat it to make sure that this is a reproducible result or that it's real. Randomization we'll talk about in a minute, but it's extremely important because you have implicit bias. You undoubtedly have some expectation, and even in the best circumstances, trying to make sure that you're doing everything exactly right with some expectation of what's going to happen, you could very subtly and inadvertently be introducing changes that can cause an effect in your experiment. So let's just consider an example. So I have a question that I'm wondering if salt affects blood pressure in mice. Now, this could be a really simple experiment to perform because I could just add salt to the water of mice and see if their blood pressure goes up. Now, what I would do in this case is I would have a population of mice or a mouse and treat their water or just put salt into their water at, say, 1%. And maybe I would allow that mouse to continue to drink that water, replacing it periodically for 14 days, and then I would measure blood pressure. Now, one problem with this experiment is that we don't have a control group. I don't know what the blood pressure is in mice. So when I get the blood pressure reading from the mouse that has been fed salt water, I don't know if that's normal or not normal. So you could compare one population of mice fed salt water to mice just fed regular water, and that's a completely acceptable experiment. Another way to think about it is, well, what if salt water affects some mice more than other mice? You could give all of the mice salt water and then compare, say, white mice to black mice, because their genetic backgrounds are going to be quite different. So in any case, you want to make sure that you have some concurrent control. And what I mean by that is that you're not taking someone else's data that reports what the blood pressure of mice is. You're doing it yourself at the same time to make sure that there isn't something funny going on that would be increasing blood pressure. A good example of this is when there's construction on campus. So construction produces vibration, and this is an example that we actually observed in a lab that I worked in. And the vibration during the day when the mice are supposed to be sleeping is very, very stressful and can actually increase their blood pressure. So you wouldn't want to produce an experiment or a set of data under those conditions and compare it to another set of data that you did 6 months ago before there was construction on campus. Now, replication is very important too. We talked about maybe you test one mouse in this setting and you see what happens if you have one mouse drinking salt water and one mouse drinking plain water. What it fails to recognize is that that one mouse is very unlikely to be representative of the entire population. In a real experiment, what you might see is that mice drinking plain water have variable blood pressure. Some mice just have lower blood pressure than others, and some have higher blood pressure than others. And the same would happen when you have an intervention like salt water. If you give the mice salt water, some of them are going to have very high blood pressure, and some of them are going to have blood pressure that looks very similar to mice drinking plain water. If you chose one mouse, what if you chose this mouse and this mouse? It would look like the salt water doesn't have any effect. So you want to produce a data set from a number of mice or a number of subjects that you can use to produce an average and then compare, use statistics to see if these two means are different. This reduces the amount of uncertainty and also allows you to quantify it. You can quantify it using statistics. You can calculate standard error or standard deviation. You learned this earlier in the semester, that a larger standard deviation means that there's more variability in the data set. So there are also considerations, like an estimate isn't really of any value unless you tell somebody how uncertain or certain you are. In other words, I could tell you what the average blood pressure was for my mice receiving salt water compared to regular water. But if I don't tell you how many mice I had and what the variability is, it's not a very meaningful statement. Randomization is intended to minimize any issues with the situation in which the experiment is performed. And when I say randomization, I don't mean haphazardly. As an example, what you may remember is that in the drug library that you tested, there were controls randomized into that set. So we used a random number generator to determine a number between, say, 1 and 24. And we replaced that tube with either colchicine or DMSO and moved that one drug into another tube later in the library. An example would be, what if you did all of your controls for your flies, say, in 1 week? And this would be really efficient because there's no confusing anything. You would do, say, all of your colchicine and all of your DMSO all on the same day and in the same week. And then in week 2, maybe you would do all of your compound. The problem with this is that what if on week 1, all of the students are there and in the room the temperature is quite high? So these flies are developing really quickly. What if week 2, we're during spring break when the lab is much cooler and they're developing much more slowly? You might then look at these larvae at the same time after giving the treatment and think that the week 2 flies are developmentally delayed or that they've died. So mixing things up is a much better design to control for any issues that might just be environmental. Other points related to experimental design that are quite important are blinding, to make sure that you are not influencing the results by having some expectation. Internal controls. This is something that we're not able to do with our flies because the outcome is death. But ideally, what you might do in the case of the mouse drinking the salt water is take the mouse's blood pressure, give it salt water, and then test that same mouse's blood pressure after drinking the salt water for 2 weeks. That would give you not an absolute number, but a percent increase, which might account for that one control mouse who has just really low blood pressure or really high blood pressure. You also want to make sure that your subjects are representative. So if you choose a small sample, it might not be representative of the entire population, because remember, what we're saying when we're testing 100 or 150 flies is that every fly in a population could be expected to have the outcome that you saw on a much smaller scale. And we need that in people too. When you do clinical trials, you're doing it on a very small number of people and expecting that that small number of people is sufficiently large to estimate what would happen in a population of, say, 350 million people. So what you want when you're designing your experiment is you want it, one, to be unbiased. The way that you can do that to produce an unbiased experiment is to randomize your samples so you're not introducing any expectation when you're setting it up, blinding so that you don't know what you're even putting in there, and also putting in your controls. Having high precision will help reproducibility. So you want to make sure that you're using the same materials every time, that the temperature is the same every time, that the compound dilutions that you're using are always the same, or if not, prepared exactly the same way. And a way to ensure that the precision is good is by reproducing your experiment. You can estimate the degree of uncertainty by randomizing your samples and by doing multiple experiments at the same time. And you'll be doing that in your independent work where you do triplicates of each of your dilutions. And then remember to keep it simple. You want to have really good answers to simple questions. Complicated questions are really complicated to solve. And you've already seen that in our meetings where you felt that you had maybe 6 files and ended up with 72 because of the controls, the triplicates, and the number of combinations that you have to include in your experiment. So let's look at this in practice. I want to highlight some research done in the Sioux lab. One, because I think it's really good research, and also it's very relevant to what we're doing. I want to direct you to the Sioux lab homepage, which if you do a search for University of Colorado, MCDB, Sioux lab, you'll find this page. And I want to highlight some things on here that you might find to be interesting and they might be useful for your project. The reason they might be useful is that Tintin Sioux is the one who identified the method, developed the method for identifying novel chemotherapies in her lab using Drosophila melanogaster. Her lab specifically is focused on understanding what kinds of things modify the effect of radiation. Now, we're thinking about this in terms of treatment for cancer, but you can also think about this as a way to protect from radiation or as a way of identifying things that might interfere with radiation when you're trying to treat a patient who has cancer. So I also want to highlight the people who are in the lab because this will hopefully help you understand the structure of one of the labs in MCDB. So Tintin Sioux is here. Do pay attention. She'll be at the poster session, so you want to make sure you know what she looks like because in the worst case, you don't want to be telling Tintin about how bad cancer is and this amazing technique that you developed because she might call you out on it. Her title is Tintin Sioux, PI. PI stands for Principal Investigator. Principal Investigator refers to the person who runs the lab. So she's the boss of the lab, and she's the one who's responsible for getting the funding and maintaining the space at the university. Her lab also has a research associate. Barb has been in Tintin's lab for a very long time. She worked at Anschutz, and she is full-time in the lab doing her own research, but also supporting Tintin's research. Shilpi is a postdoc. Postdoc refers to a position that's secured after you receive your PhD. In the life sciences, if you're planning on staying in academia or doing research, most positions are going to require a postdoctoral fellowship. And this is a position that you hold for anywhere from 4 to 6 years usually, and it just provides more independence. And this is the time in your academic career anyway when you really want to be publishing like crazy, because after this position is when you start applying for faculty positions. Nate is a postdoc, but he works at Tintin's company. So her company, which allows her to move things even closer to clinical trials, is in her lab. It's very small and has only 2 people, Nate and one other person who's not shown here. And then what you see here are undergrads. All labs in MCDB have a website, and I would encourage you, if you're interested in finding a lab to work in, look at their website and see how many undergrads do they have. Do they have none? Do they have 10? This will give you a sense of how supportive they are to undergraduates and how likely it is they are to take undergraduates in the future. Tintin has 5 undergraduates, and this is a large number of undergrads. They stay in the lab for anywhere from 1 to 3 years, so you can expect a high degree of turnover, but I would pay close attention to the labs that have a lot of undergrads in them. Now, the projects are also listed here, and you can see that we are here. This is our project that looks at the effects of radiation combined with compounds on the survival of larvae that are susceptible to radiation. But she also looks at the general mechanisms of radiation damage. In other words, what happens when the cells are exposed to radiation. We know that it causes double strand breaks, but then how do the cells repair? How do they know that other cells are missing and they need to start dividing? The publications are interesting because you'll be able to go there and see a list of relevant publications to your work. So don't forget that the work that you're doing right now is directly related to Tintin Sioux's work, so you might want to take a look at this list of publications and at the very least read the abstracts to see what's happening in that lab. Now I have an example here that you read an excerpt from. I gave you 2 figures to highlight how her research relates to our research. And I want to mention here that Anthony Edwards was an undergraduate in the lab. And what I'll do here is I'll walk you through this paper so that you can see how she presents the research once it's done. Now, the introduction section of a paper is usually anywhere from 3 to 5 paragraphs. And the intention of the introduction is to introduce the problem, to sell the project that you're writing about, and also to sort of hint at what the result might be. In Tintin's introduction, what she did is she comes right out of the gate. Now, this is the elevator talk. You have 30 seconds, basically, to tell somebody why they should be paying attention to you. And I think that in this paper, Anthony Edwards and Tintin Sue do a really good job right out of the gate saying what they need to say. Cancer is bad, treatments aren't great, we have a solution. So it really gets a reviewer or somebody who's potentially interested in her data to pay attention. The second point that they make is that they developed a technique, this drug screen, in Drosophila melanogaster. They tested it and published it in a previous publication, and they found something cool. So you can refer to this other publication to see what happened, but you can also understand a little bit about the approach that she's going to be presenting in this paper. Then what she talks about is using that same technique. They found 13 hits. Some of them were already chemotherapies, which is super exciting because it means that the approach works. But one of them hadn't been described as a chemotherapy ever in the literature. And that's the thing that she's presenting on. So you can see how clean this is. Cancer is a problem. We have an answer. Check it out. It works. In the methods section, you'll notice that her methods are very similar to what we're doing. She used third instar larvae. She irradiated at 4,000 rad with the aim of killing 50% of them and treated with metanosol. What's interesting is that she also used human cancer cells that were also irradiated at 4,000 rad and treated with metanosol. And this is sort of the next step in our experiments. We're doing number one. And then after number one, the next thing you want to do is drive it a little bit closer to home. And that would, the easiest thing to do is to use human cancer cells. Then maybe you would inject those human cancer cells into a mouse, and you would try to understand a little bit about the biochemistry. So let's walk through the data. Whenever I'm approaching a paper, there are usually more graphs than you care to look at. So it's important to have an idea of how you're going to approach that graph every time you look at it, because it's going to make things more efficient. So in this graph, I know right now that they are testing wild type something. It turns out that they're larvae. She's looking at the percent survival to adulthood, and that's what we're doing too. We're looking at the percent of larvae that survive and eclose. Remember, and this was something that a lot of people didn't get correct on the midterm, partial eclosure is alive. If a fly is able to start penetrating the pupa, the pupal case, and start to get out, it's an adult. Even though it's still dead, if it's halfway hanging out, it was considered a live adult. On the x-axis, they're looking at the concentration of drug. So before I even look at the bars or the error bars or anything else, I'm going to say to myself, oh, they're testing larvae, 2 different genotypes. They're looking at survival, and they're testing increasing doses of the drug. Then I walk through the genotypes. Oh, wild type, without anything, survived the same as P53 mutants. P53, as you know, is the gene that's often mutated in cancers. So I know that those flies that are mutated don't have a problem at baseline. Then I look at what the bars are doing. Both of the colored bars are going down with increased treatment with metanosol. So what that tells me is that the increasing dose of the compound is more toxic to both genotypes. So that's a kind of a cool result. So then what she does is she says, okay, well, a lot of the flies end up dying because they're exposed to this drug. But what happens if you combine it? So what she did here is, here it says P53. These are wild type flies that are exposed to 0 radiation or 4,000 radiation, increasing doses of metanosol, and how many survived. So this here should be wild type flies. So she's looking at the wild type flies again. Wild type flies. Wild type flies are sensitive to metanosol by itself. So if you give the flies increasing doses of metanosol in white bars with no radiation, they start to die. These same flies, the wild type flies, are more sensitive to metanosol plus radiation compared to metanosol by itself. So that's kind of cool. That means that you can combine radiation and metanosol and get even more death than the drug by itself. In the third experiment, here she's looking at the p53 mutants. And here I look and I say, okay, these are just the same things. This is increasing dose of drug, survival, survival, radiation or not, radiation or not. And here she is, the p53 mutants. The p53 mutants, when they're exposed to both the combination of metanosol and radiation, they're very, very sensitive to it. In other words, a very low dose of metanosol is effectively killing all of them. So this is really exciting. It means that this might work in a cancer cell. What she does next is she compares cancer cells, which are HCT116 cells. These are colon cancer cells that have no mutation in p53, a mutation in p53. They look at increasing doses of metanosol again, and they look to see how many are surviving after some amount of time in treatment. So what I look at first is, okay, they're giving metanosol and both of the colored bars are going down, meaning that both non-mutated and mutated human colon cancer cells are responsive to metanosol. So that is super, super exciting. It means that these might be responding very well to the compound and that it represents a potentially interesting new drug. In the discussion, what she does is she sort of reiterates what she already told you. We developed a technique. It's cool. It works. But what she's saying here is, and this is what defines a discussion, is things that you just want to put out there. What she's saying here is that, yes, this approach can identify lots of different things, but probably not everything. And in the next section, what she talks about are the limitations. She's acknowledging that she can't identify a mechanism, but she's also saying, hey, if you're going to do this again, these are the kinds of things you would do next. Now, in looking at this, I thought it was interesting because this is sort of the data that you're going to be producing. So what if we put it onto a poster? So I just put it onto a poster, and this is what it looks like. I took their abstract from their paper, and I put it into an abstract section. This summarizes the entire poster, including the results. I put part of their introduction into the introduction section, and then because it seemed to me that a lot of their emphasis was on the approach itself, I put a picture of the approach because this is what they're actually reporting. I wrote down their hypotheses. I wrote down their methods with some pictures, which are from the discoverylabs.org website. You're welcome to use them. And I also put their results. I only put on here the results that they got using larvae. This is a lot. These results are, you might only have one graph, and some of you might have no graphs. Maybe you'll just be reporting hypothesized results. And on a supplement that you'll hang off to the side, you'll have results that you obtained after printing the poster. There won't be any point deduction for this. This is very commonly done. And then in the conclusions and future directions, this is where you talk about what it means in the context of the field. And here in the future directions is where you talk about what you would do next. In the acknowledgments, we'll give you a list of people that you should acknowledge, but you can really acknowledge anyone who you think helped out with the project. And then references. You'll be required to include at least 4 or 5 references, but you can put them really small and they don't need to be in text. In other words, you don't have to have numbers or author names anywhere in your text indicating where to find this information. So that covers what we missed because of the snow day. You can ask me any questions that you like. Again, the midterm is going to be on Wednesday, April 17th during our class time, and there'll be a review session on Monday.